Hello students, welcome back to my channel. As you can see that today I am going to discuss on the next topic Megasporogenesis. Last day I had discussed microsporogenesis. Macrosporogenesis, you had seen that there is a formation of the microspore, that is the male gametophyte. And now megasporogenesis, formation of the megaspore, that is the female gametophyte. So male gametophyte formation and the female gametophyte formation, these two are the phases of the pre-fertilization stage. Okay, so we are continuing the next part of the chapter, sexual reproduction in flowering plant. Before going to this major topic, that is the megasporogenesis, we shall recall some very common facts on female reproductive parts of a complete flower. You know, biology, it depends on the visualization, and visualization, it develops from the diagram. So in biology, diagram is very important, but not artistic diagram. You can go for a conceptual diagram where you have to label the parts properly in the proper position. In CBSE marking scheme, in board exam, they emphasize on the labeling, not on the very artistic diagram. So these are the parts you have read in class 10, that is the stigma, this is the style, and this is the ovary. You know all this, very common. And inside the ovary there is a structure. We can show it in different way. And is in class 11 also you have read that there is a structure and we name it as the ovule. Ovule, which remain attached with the ovarian wall in different pattern, and that we had read in class 11 as the term placentation. Okay. So today we have to start from this ovule. Okay. So my next topic today now, which is related with megasporogenesis, that is the ovule. We shall start with ovule. Students, be very careful that this ovule has another name, that is the megasporangium. Megasporangium. Okay. So again, before going to the, we will now discuss first the structure, and as usual in microsporangium we had discussed the ploidy. So here we should be very clear what is the ploidy, and ploidy is 2n, that is the diploid. And so this structure is a sporophytic body of the flower. Sporophytic body. Okay. So let us draw a very simple structure to see the parts of the ovule. Okay. So let us start with this. So here I am starting the diagram portion of the style I am drawing, and then I am drawing the ovary so that it will be very clear to you how is the ovule. Now I shall draw the ovule. So in this way, very very simple diagram I'm showing you so that it will be easier for you to understand the parts of the ovule. Okay. So this is the part, one part, and this is the continuation of this. Just now I will label all this, and there will be a very big cell I am drawing here, and the rest of the part I am filling with this. Yes. So let me fill. So now let me label all these parts. First, this small opening, okay, this hole. First, you remember that this is ovary, hmm? And this whole diagram is of ovule. So this small opening is called micropyle. Then these two layer is called these two layer oh, are called integuments. Integuments. Okay. Now this portion which I have filled, it is actually a cellular portion, and this is termed as nucellus. All the cells are there, but one cell is very significant here and bigger in size in comparison to other cells, and this is called the megaspore mother cell, MMC, and this is the funicle, and this part, merged part here with the funicle, is called hilum. Okay. So when you are asked the different parts of the megasporangium, these parts, then what you will write? First, integument, or you write micropyle. What is micropyle? A small opening, very small opening for the entry of the pollen tube. Okay. Number 2, integument. Protecting layer. Okay. Then number 3, what is there? That is the nucellus. Cellular structure. which gives what? Nourishment. Nourishment. Okay. Then we will go to the MMC. That is the megaspore mother cell. Which is the precursor of megaspore. Precursor of megaspore. And then is coming the funicle stalk like structure for the attachment of ovule. So students, ovule is attached to where? On ovarian wall. And then it is coming the hilum. Merged part of merged part. Or you should write in this way. Part of integument. Merged with funicle. So this is all about the structure of the megasporangium, the sporophytic body. Next we are going to the megasporogenesis. How megasporogenesis occurs from the sporophytic body, megaspore mother cell. As from the name, mega means big, and the spore, big spore, genesis means formation. So what we will write? What is megasporogenesis? Formation of megaspore from megaspore mother cell through. Now you know what is a division? Meiotic division. That is called megasporogenesis. So let us write the flow chart then. What should we write? Now I will write MMC. Question comes on MMC full form. Either MCQ or whatever 1 mark, 2 marks questions, they can add this MMC. Now MMC will undergo what? Meiosis. Okay. So now there will be how many megaspore? 4 megaspore. 4 megaspores. Okay. So how it is occurring? Let us see. Suppose this is the MMC. Okay. So we can draw here also, that is better. This is MMC and it is giving rise to 4 megaspores. So these are 4 megaspores. Among these 4 megaspores, it is very important the 3 degenerate. 3 degenerate. Okay? Degenerate. So how many is existing? 1. Only 1 exists. Okay? And this one ultimately gives rise to the embryo, white color, embryo sac, which is called the male gametophyte. Male gametophyte. Okay. So again, to recall the previous diagram, let me make the conception clear. So this was the ovary. There we had made the ovule inside, I think with red color. Yes. This is the ovule. Okay. And whatever is forming inside the ovule from the megaspore, that is the embryo sac. Okay. So this is the embryo sac will form from one megaspore. This is the embryo sac, and that is the male gametophyte. It is forming from how many megaspore? 1 megaspore. 3 are degenerating. So this type of development of embryo sac from 1 megaspore is called, very important, monosporic development. It is called monosporic development. If you are asked what is monosporic development, you can say this one. Now it is very clear. Okay. Now next topic we are going to discuss the structure of embryo sac, because the embryo sac is not so simple. It is an oriented structure. Okay. So structure of embryo sac. Here it is female gametophyte. Female embryo sac, that is the female gametophyte. So now let us see the structure of the embryo sac. Structure of the female gametophyte. Or the embryo sac. How it is developing first. See. Megaspore. Embryo sac, what is the ploidy? Haploid. Megaspore, it is haploid. Now it is undergoing mitotic division. No meiotic. What is the division here? Mitosis. Then it will form 2 nucleated Megaspore. Then it will form after another division, consequent division of mitosis. Ultimately, it will form 8 nucleated structure. Okay. So, let us draw this one. Suppose this is the megaspore and 2 nucleated means how many nuclei are formed? 2. Then ultimately it is forming 8. You can see 8 nuclei. So we can see here the whole process which we can see, that is only the nucleus is dividing. So it is called strictly. It's very important. This is strictly free nuclear division. What does it mean? That is no wall forms up to 8 nucleated stage. Okay. Now from the after this 8 nucleated stage, the walls are forming. Clear? So be very careful that no wall forms before 8 nucleated stage. So let us form wall around each nucleus. Now if I count all the cells, we are getting 1, 2, 3, 4, 5, 6, 7. So here I can write that the embryo sac is now 7 cell stage. Okay. And how many nuclei? 1, 2, 3, 4, 5, 6, 7, 8. So 8 nucleated. So the question comes, describe the development of the 7-celled 8 nucleated embryo sac. Okay. Now let us label all this. These 3 are called antipodal. Antipodal, I shall give you the idea. Which one you should say antipodal? For giving this idea, I have to draw this one, this ovule. I had drawn ovule with this. So whatever is happening, first let me show you that ovary. This is the ovary. Again, this is the ovary, and this was the ovule. Whatever was happening, that is in the ovule. And inside this ovule, there is the embryo sac. This is the embryo sac. So, if this is the micropyle, of the ovule. If this is the micropyle, then the antipodal cell in the embryo sac will be just opposite to micropyle. Okay. Now it is clear. So I have labeled antipodal cell where just opposite to the micropyle. Chalaza end. Okay. Now this is what? Central cell. Or polar nuclei. This one is very significant, that is the egg. And these two are synergids. So this is the complete embryo sac or the female gametophyte. Now I will emphasize here one point, that is the synergids. Synergids have much importance because these are associated with filiform apparatus. So let me show, if this is the embryo sac, this is the embryo sac, and if this is the egg cell, so the synergids are associated with filiform apparatus. This one, filiform apparatus, and these are the synergids. So this we say filiform apparatus. Why it is important, significant? Because it guides, the question comes, it guides the entry of the pollen tube through micropyle. Okay. So today we had covered the formation of the female gametophyte. So pre-fertilization stages are over. Next we will go to the fertilization stage. Before that there is one connecting stage, that is a pollination. So we will meet again with that topic. So today up to this, please ask me questions if any query is there. And I teach like a tuition class, so that every details of NCERT line by line I want to make clear, so you get the conception very clear and easy to answer the questions. So whatever query you have, please tell me in the comment box, so that I will be also updated, your problem also will be solved. Thank you, students. Let us meet next day.